Okay. Hi, everybody. Good morning. I see some new names. I see Kirti Vasan, Meena. All these are new names I'm seeing. Kirti Vasan, are you new?
Yes, sir.
Okay. This is the first class you're attending for chemistry.
Yes, sir.
Okay. So how about physics and math? Have you joined there also?
Yes, sir, but not yet. I'm not having the class.
Okay. Okay. Understood. Meena, how about you? I think she is now only connecting the audio. She'll not be able to speak, I guess. Okay, no issues. Whenever she gets ready, we'll do it. Right. So I think we can straight away start the class. Let me share my screen. Okay. So we were seeing biomolecules. Last class I just gave you introduction. Right. Right. Fine. So regarding carbohydrates, you have to know what are monosaccharides, what are disaccharides, what is a glycose. Okay, let me tell you what all you have to read. Okay. Monosaccharides. Right. I have disaccharides, polysaccharides. You have to know examples of all of them, right? So disaccharide example is sucrose, right? For this you have to know glucose, fructose, right? And for polysaccharide, what you have to know, Kana? Like what are the structures you have to remember? For polysaccharides example?
Sir, starch, cellulose,
Starch, then
cellulose.
cellulose. Okay, I think these two are enough. Okay, starch and cellulose. Cellulose, I think starch, I think you have two things, right? Amylose and amylopectin. Right, starch has two components in it, amylose and amylopectin. Again, how they are different, everything you should know. Right, so first of all, their structures: glucose, fructose, sucrose, starch, cellulose structures. I mean, obviously starch and cellulose structures you cannot remember because it's very big, right? But you have to know what is there in starch, what is there in cellulose, what are the monosaccharides. Both of them are made out of glucose only, right? Both of them are made out of glucose only. So many glucose units will come together to make starch and cellulose. Different kind of combination, right? Okay, so these are all the structures you have to know. And after that, obviously, for the mono and disaccharide, you have to know two structures. One is the straight chain structure, right? And the other one is your cyclic structure. Both the structures you should know. Okay, the straight chain structure, cyclic structure. Again, in cyclic structure, the glycosidic. I'm sorry, I'll write again. Glycosidic linkage or glycosidic bond, you should know, right? How the glycosidic bond is formed, between which two carbon atoms of glucose and fructose it is getting formed, right? And also for these two, you have to know what are reducing and non-reducing sugars that we saw last class. Non-reducing sugars, which are these and how do you identify them? I think I told you reducing sugars will have your hemiacetal. Right, hemiacetals we know. Hemiacetal means what? One carbon atom having OH, the same carbon atom having OR. This, if you see, then it is going to be a reducing sugar. Right, and for glucose, you have to know all the reactions. Right, for glucose, you have to know all the reaction. Mild oxidation, I can even write mild oxidation, that is your bromine water. Right, and you have to know strong oxidation, and that is your HNO3, right? Then I think you will have the ammonia derivatives. I think it is ozone formation, right? Ozone formation, you basically go for hydrazone, you add it to the carbonyl compound, that is your this thing, you're gonna get ozone, right? So like this, whatever reactions you have in the book, right, in your NCERT textbook for glucose alone, please get to know about it. The names, obviously, when I say reactions, you have to know what is the reactant, right? What is the reagent and what is the product. The names also are all very important. Name as well as the structure, right? All these are very, very, very important, right? And also, especially in sucrose, you have to know this very important reaction, that is my inversion of cane sugar, right? And I know this also is called as what? Mutarotation. Okay, all these things are thought things you have to study about by sugars, carbohydrates. Okay, all these are the things you have to study about carbohydrates, right? All of them should be in your fingertips. Very, very simple. Okay, right? Any questions anybody has regarding carbohydrates? Okay, fine. I assume that everybody is okay, right? So today we will move on to proteins. Okay, right? We will move on to proteins. Last class, I think I told you about how a biological system works, right? A biological system works on this particular, say, particular baseline, right? What? First, I have something called as the DNA. DNA gets converted to RNA, and RNA gets converted to proteins. We all know this. Okay, DNA to RNA is what is called as transcription, right? RNA to proteins is what is called as translation, right? And I can even have the RNA getting converted to DNA, and I know that is my RT. RT is what? What is RT? What is RT, people?
Reverse transcription.
Reverse transcription. Transcription is DNA to RNA. Reverse transcription is RNA to DNA. Okay, right? Right. So now actually I want to tell you this point, right? Generally, right, when you go to Big Bang theory, right? When you go to Big Bang theory tells how Earth came into existence. Correct. What do they say? They say that it was the proteins which were first formed. Right? It was the proteins which came into the first place. Then only the other things came. Right? Now, please understand, when I say proteins, I told you in the last class itself, proteins have different roles in our body. Some protein can act as enzymes. And I know what are enzymes. Enzymes are catalysts. Right? And basically, for all these reactions, all these are chemical reactions. Okay, transcription is a chemical reaction. Translation is a chemical reaction. Right? So for all of these reactions, you need some catalysts. Right? And that catalyst can be proteins. Right? For example, if I have to convert DNA to RNA, right, I need to have something called as polymerase. Right? And this is nothing but an enzyme. Enzyme means what? A protein. So what do you understand? Proteins are formed, and those proteins itself act as catalyst for some other reaction. Okay, right? So like this, there are so many things. For example, I will be able to convert your glucose C6H12O6 to carbon dioxide and water. Right? Now here, the enzyme used is glucose dehydrogenase. You don't have to remember anything. Not this name polymerase, not this name dehydrogenase, nothing you have to remember. All I'm trying to tell you is these are all the normal chemical reactions happening in our body, and these reactions are catalyzed by our biological catalysts, and those biological catalysts nothing but enzymes. And what are enzymes? Enzymes are nothing but proteins. Okay, right. Fine. So why did I start this is because I told you that during the Big Bang, it is predicted that proteins are the one which were initially formed, right? And after that came DNA and RNA, right? But the thing is, from this dogma, I know this is what is called as central dogma of biology or of molecular biology, right? So if this is going to be followed, how is it that from proteins I would have formed DNA, RNA, right? So this is a very good debate you can have. What scientists claim now right? What scientists claim now is that it is the RNA which came first, not DNA, not proteins. It is the RNA which came first. And you can easily understand why. Because from RNA, I can easily form proteins, following central dogma. And by reverse transcription, RNA can also form DNA. Hope you understand what I'm trying to say. So people are still having debate out of these three, which came first. Some scientists claim that it is proteins, but now scientists are moving towards RNA. They say that RNA is the one who's getting first produced, and from that, the life evolved.
Sir, why not DNA, sir?
Why not DNA? DNA, okay, fine, that's a good question. Now, the thing is, DNA is a very complex structure, okay? We'll study about DNA in a while, right? Now, DNA is a double stranded structure, very stable when compared to RNA. When I compare the reactivity of RNA and the DNA, right, RNA is more reactive in the sense that DNA is like little inert. It will not undergo reactions that easily, right? But RNA is little unstable. Compared to DNA, RNA is unstable, so it can undergo a better reaction. Say better reaction means it can undergo reaction faster. It is more reactive than DNA. That's the reason. Okay, right. So yeah, understood. So these are all the things you can know for extra knowledge. Okay, now moving on to proteins, right? Now proteins are a polymers. Proteins are polymers. We know what are polymers, right? Just like how you have polysaccharides. What is what are polysaccharides? So many monosaccharides. For example, in starch, so many glucose units comes together, make some bond and make a polymer, and that polymer is what is called as polysaccharide, right? So here also some functional unit, one unit will be there, and that unit will be coming together, making a very big molecule called as protein. Okay, now what you have to understand is what comes together. What are the monomers? That is the exact term I should use, right? So many monomers will add and add and add to form the polymer. For example, this is irrelevant to this. For example, if you take polythene, right? Now you see it is polyethene molecule. So how do I write it? So n moles, not 2 moles. N moles of CH2 double bond CH2 will form CH2, CH2, a big molecule, n times. CH2, 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 is gonna keep on moving like this. Okay, this is my monomer. And this is my polymer. You understood, right? So many monomers, single unit, will come together to form a polymer. Right? Now, same thing only is going to happen in case of proteins. I told you proteins are polymers, right? So many monomers will come together to make this polymer. Now we have to understand what are the monomers, right? Now we have to understand, people. Monomers for proteins are, I would say, alpha amino acids, right? What are the monomers for proteins? Protein is a polymer. It needs a monomer. What are the monomers? The monomers are alpha amino acids. Okay, right. What are these alpha amino acids, people? Any alpha amino acid will have this structure. It will have a carbon, right? It will have an NH2 attached to this carbon, and I will have COOH attached to that same carbon. Right? Now you understand, this is your carboxylic acid. This would have been your alpha carbon, right? This would have been your alpha carbon, and that carbon is having NH2. That's why we call it as alpha amino acid. Or it's very simple, same carbon atom having NH2 and OH is what is called as an alpha amino acid. Right? Now, please understand, people, depending on what are these two bonds, what is there can be some alkyl groups, right? R, some R dash, R double dash. It can even be some functional group present, right? So, but all you have to look for is this, right? The alpha amino acid part. Okay. Now, please understand, people, in our body, for humans, right, I require 21 amino acids. might be higher. I think 21 is the correct number. I think it's 24 or something. Anyways, some 21 or 24 amino acids are required for me to form different kinds of proteins. Okay, right now, how are these amino acids coming together? First, you have to understand. Okay, the monomer is an amino acid, right? Next, you have to understand is how these monomers are attached covalently. Right, so many monomers, so many amino acids have to come together to make a protein, right? Okay, now it is very simple, people. Say I will take one, okay, right? I will take, for example, there's something called as glycine, right? Glycine is an amino acid. Okay, now glycine is very simple. Okay, it is CH2, no, C, NH2, COOH, H, and H. Both of these are H, these two. What am I writing? This is the simplest amino acid. Okay, my glycine is the simplest amino acid, and also this is the only amino acid which is optically inactive. This is the only amino acid. I told you there are 24. Again, I think it's 24. Okay. So out of the 24 amino acids, right, which are inside our body or which we take through diet, we'll come to that point. Which amino acids are inside our body, which amino acids you have to take into diet, right? That we'll see later. But either through the diet or in your own body, there are 24 amino acids. Out of these 24 amino acids, only glycine is optically inactive. Now you can see why, right? You see both these are hydrogens. Right now, what is the criteria for being optically active, Kana? What is the criteria for a compound to be optically active? Can someone tell me?
So the chirality and the asymmetry.
What do you mean by chirality?
When a carbon is attached to 4 different groups.
Right. When the first condition is chirality, and obviously second condition is symmetry. Right. Both these should not be there. I mean, chiral carbon atom should be there, and symmetry should not be there. Right. Now you see the first criteria itself is gone. Now this particular carbon is not attached to 4 other groups. Now, how do I write OH2? Oh, this is NH2. I'm sorry. Okay, now one NH2, one COOH, but you see that in glycine, I have 2 hydrogens. That means there is not 2 different groups, right? So, but if you look at other amino acids, you will see that there are going to be different groups here. Here there'll be one group, there'll be another group. So all the other amino acids are optically active, right? But glycine alone is the one which is going to be optically inactive. Okay, right. Now, before moving on to see how a bond is formed and all that, right? So please understand that this amino acids can be it can be divided into 2 types. One are what are called as essential amino acids, right? And the other ones are what are called as non-essential amino acids. Okay, essential, non-essential means it's not like I require them and I don't require them. No, essential means they have to come from the diet. Okay, we have to take it in. Our body cannot produce our own amino acids, right? Some amino acids can be produced by our own body, right? Some amino acids cannot be produced by our own body, so you need them still, right? So you take it from your diet, right? And the non-essential amino acids, which you do not require from outside, it is produced by the body. Okay, it is produced by the body. Okay, now which and all are essential, which and all are non-essential is really not the topic. Okay, but you have to understand that there are some amino acids should be taken to the diet, and there are some amino acids which can be produced by our own body itself. Okay, but all these 24 are required. Okay, right. So let me go to Google and show you what are all the different kinds of amino acids you can have. You do not have to remember the names. I mean, remember names, yeah, you can remember, but for now, you don't have to remember the structure. Okay, now let me show you. There's something in the chat. 8 chats are there. What happened? Sir, today there should be a question paper discussion. Okay, paper discussion. So is that okay. Fine. I'm so sorry, guys. You want to do paper discussion, but we didn't even complete the chapter. When is we have to complete the chapter? Then only we can talk about revision and all. Right? This chapter is not even completed. What do you suggest, people?
Actually, last class you discussed only 12 questions, I think so.
I know.
Haloalkanes and solid state, sir.
I understood. This week, did you write any test?
No, sir. Hereafter only you're going to write test. Biomolecules and P block hereafter only, sir.
Oh, next chapter is biomolecules and P block.
P block, yes, sir.
So yesterday we
Then we have to do biomolecules. Right? We didn't even complete.
Yesterday we wrote alcohol, phenol, and solutions.
Oh, you wrote yesterday alcohol, ethers, phenols, and solutions.
Yes, sir.
How was the question paper? Was it okay?
Moderate, sir.
It was okay, sir.
Okay. Were there any questions which came out of the book? I mean, out of the syllabus?
Actually, 2 questions are repeated, sir. I said it in the center, sir. They said we have to write any 1 question. That's enough. They said,
Okay.
sir.
Okay. Question by question we have to do. Yes, that's a very important point. And then we have to complete this chapter also. When is your next test? Next epsilon test, when will you write? Next Sunday or next Saturday?
next Saturday.
Next Saturday. Okay. So if that's the case, we have to complete this chapter and I have to do a question paper discussion also. Fine. So for today, we'll complete this chapter. We'll try to complete this chapter. It's very little things are only there, right? Or else maybe one more class in the weekday. But before you go to the exam, we will try to come. We will complete, not try. We will complete this chapter for sure. But question paper discussion, we need to have an extra day. Actually, that is very important. I have to discuss this with you. What can be done? Okay, we'll not waste time. Today we'll continue. Okay, today we'll continue with biomolecule. Let me complete it. I'll try to complete it as much as possible, right? And then in weekdays, some weekday, we will have some class. Okay, we will have a class for question paper discussion. And don't worry, we will do it. We will do a question paper discussion for sure. Okay, right, fine. So as I told, amino acids can be essential amino acids and non-essential amino acids. And now I'm going to show you the amino acids. What are the different amino acids? I want a list of amino acids. Come on, give me a proper structure. Give me, is this fine? Little more clear if I can get. Okay. No. Some good image is what I'm looking for. Better. Let me download this. Okay, right. So I think everybody is able to see what is there.
Yes, sir.
Right. Okay. Now we are. I already saw glycine, right? Okay. So glycine is basically same carbon atom having NH2 and C over H. Okay. You see that these two are H2s. Both these carbon atom will have H2. Okay. Now you see an alanine. Alanine is basically CH. If I can write it, it is going to be C NH2 C over H. There is a H and there is a CH3 here. So basically you see that H and CH3 are the other two groups, and that is my alanine. Okay, now you see so many things will be there. Now in this particular line, right, in this line, that is glycine, alanine, valine, leucine, and isoleucine, all of them contain the side chains. Side chain means I'm talking about the other two groups, right? The two more bonds are left for carbon, isn't it? Other than NH2 and C/H, there are two other bonds left out. These side chains are hydrocarbons, just hydrocarbons. You can see, right? This is CH2, this is CH3, there's another isopropyl group, some other group. All these are hydrocarbons. Okay, now when you go to the second line, right? Now, second line consists of some kind of polar groups. Now you see that in aspartic acid, I have a COOH, right? COO minus basically. Same way in glutamic acid also, you have a COOH, right? In aspargine, you see that there is a amide. This is an amide, right? So all the things, whatever you see in these things, right? All of them contains what? All of them contains some kind of polar groups. Okay, and if you go to this one, right, if you go to the other one, that is the third line, you will see there are aromatic rings. You can see that green colored things are going to be aromatic. It's given here. Like, okay, if you look at this chart, red means aliphatic. You can see all these are reds, all these are aliphatic, right? Green means aromatic. You can see aromatic ring is present, aromatic ring is present, aromatic ring is present. Right, and amide is yellow. You can see not yellow, orange, right? This is an amide. You can see this is also an amide, that is an orange color, right? Hydroxylic means OH. Where is serine? Serine is what I'm looking for. Serine is here. Serine will have OH. Serine is very important. Okay, it's very, very, very important with respect to biochemistry, but don't worry, you don't have to go into the details now, right? So now you have OH. So you see, right, different amino acids are there. And all of these amino acid structures are different, right? And their structures are different because they'll have different roles to play in a protein, right? They will have different roles to play in a protein. Okay, right. So we understood what are amino acids, what are essential amino acids, what are non-essential amino. Out of these, some of them are essential, some of them are not essential. Okay, don't worry about it, right? And you see, each of these amino acids are given a 1-letter code. Now you can see for glycine it is G, for alanine it is A, for serine it is S, for threonine it is T. Like that, 1-letter codes are also given for the amino acids. Now it is just to makes things simple, because in a protein, in one protein, I can have 1,000 amino acids. Okay, just giving you a number. In one protein, I can have 1,000 amino acids. Okay, so for all these 1,000 amino acids, you cannot write these names like this, right? So I will just represent the protein like this: A, G, T, S, P, something like that. I'll write. That means what? If first is alanine, glycine, threonine, serine, proline, like this, I'll be able to write 1-letter codes. I can write 1,000 letters like this. Okay, right? So that is what is the importance of these single-letter codes which you have. Okay. Now this I wanted you to understand. And next thing is how a protein is formed. Now we understood that these are functional units. Amino acids are functional units, right? Now these functional units have to come together, right? They have to come together to make a bond so that proteins can be formed. Okay. Now all you have to understand is, people, let me take any two amino acids. Let me take glycine and alanine. Okay. Let me take glycine and alanine. Right, now we'll understand how this glycine and alanine will come together to make a bond. Okay, now you see that glycine's structure I know is C, NH2, COOH, and H and H. This is my glycine, right? Now look at alanine. Alanine is, I'll write H2N C COOH, a H and a CH3. Okay, right. Now what you have to understand is, people, in a protein, right? How proteins means it is a mixture of amino acids. So many amino acids coming together. how two amino acids are linked, right? How two amino acids are linked is by something called as a peptide bond. Okay, how amino acids are going to come together to make a bond? What is the name of the bond? It is called a peptide bond. Right, so that is why we call proteins as polypeptides also. That is why we call proteins as polypeptides, because they are made out of peptide bonds. Okay, they are made out of peptide bond. Now, what exactly is a peptide bond, people? A peptide bond is nothing but an amide bond. A peptide bond is nothing but an amide bond. We know what is an amide. Amide is something like this: CONH, like this and like this. That CONH, if you form, that is what is called as an amide. And in biology, we call this amide bond as a peptide. Okay, we call this amide bond as a peptide bond. Okay, now how do you form this? Let me show it here. Right, let me write one H and another H like this. Okay, now very simple, people. From one amino acid, you remove a OH, and from another amino acid, you remove a H from nitrogen. Okay, in one amino acid, you remove OH from COOH, and from another amino acid, you remove H from nitrogen. Right, so if I remove H2O, basically H and OH will put together minus H2O. Okay, now you can easily form the amide bond, right? Let me write it like this. Can I write it like this? CH2, this is this carbon, CH2, and what is there? That is having a NH2, right? And this is having a C double bond O. I'm talking about this one, C double bond O. And that is attached to this nitrogen now. That is having an NH, right? And that is attached to what? CH, COOH, and this I have a CH3. So COOH is here, CH3 is here, CH3 is here, COOH is here. Now you see all that is important is this one. This is my peptide bond. Okay, this is my peptide bond. Not only glycine and alanine, any two amino acid, if you want to mix them, if you want to make them bond together, in all of these amino acid bond formations, you will see that there is going to be a peptide bond. Okay, you will see that there's going to be a peptide bond. Again, this can continue. Say, I can have serine coming here, right? I can have a serine coming here, and between this and this, I can have a peptide bond again. So this can grow as much as big as possible. Okay, this particular chain can grow so much bigger through the peptide bonds. Okay, right? So that is what is called as your peptide bond, right? How proteins are formed? Proteins are formed from amino acids coming together. How amino acids come together? It comes through a peptide bond. What is a peptide bond? An amide bond. And how do you form an amide bond? Removing OH from COOH of one amino acid and removing H from the nitrogen of another amino acid. It can be same amino acid also, not a big deal. It can be glycine and glycine, alanine and alanine, serine and serine. All these are okay. It can be different amino acids also. Okay, right? Fine. So that we understood, right? Now, if you go to the structure of protein, right? If you go to the structure of protein, right? Now, at what level are we looking at is the question. Okay. Now, if at all I see very superficially, right? I see superficially, this is the kind of structure you will get for a protein. Nothing you'll understand basically. Okay. See, let me show you. Right. Say, I want to show some protein, right? Some proteins, say alcohol dehydrogenase. Now, see what kind of structures you're getting. Are you able to understand anything from here? Right. This is the kind of structures you will have for proteins. Okay. Or a better picture I'll show. This is an X-ray. This is done through an X-ray. Okay. X-ray. It's called X-ray crystallography. Okay, say you see this is human liver alcohol dehydrogenase, which is nothing but a protein. Right now, all you see is some kind of ribbons and wires, something attached. Like this is what you see. Right. So this is one level of seeing. Right now, I can zoom into this and see what exactly is happening. Right, but that here I cannot show you. If I had an animation, yes, I can show. Right. So what you have to understand is this is the kind of complex structures proteins can have. Right, and these are nothing but bonds. These are every inch of this particular structure is a chemical compound. Right, and all of these are amino acids only. Amino acids somehow come together, so many amino acids, and they form this kind of structure. Okay, and this particular structure, what you're seeing is what is called as quaternary structure. Right, this what you're seeing is what is called as quaternary structure of protein. Right, this is very superficial. This is the data we'll be able to get from the machines. Okay, fine. Anyways, moving on. Right. So as I told you, what level are you seeing is very, very important. Right. Now, there can be primary level, secondary level, tertiary levels, and obviously the last one is quaternary, and that's what you saw now. Okay, whatever the end structure you're seeing, the superficial structure, this kind, whatever you have like this, that is my quaternary structure. Right, so structure of protein can be studied at different levels: primary structures, secondary structure, tertiary structure, and quaternary structure. Okay, right. Now what you have to understand is, what do you mean by a primary structure? Now, primary structure is the in-depth structure, in the sense it is going to give you the sequence of amino acids. Right, now wait, what do you mean by this? Right, say I write alcohol dehydrogenase now. You see this, let me view it. Okay, now you see this is the primary structure. Right, now again it's going to be very complicated. Now you see this THR is 309. Right, TRP is basically what? Tyrosine. Right, MET is methionine. Right, CYS is basically cysteine. Right, HS is histidine. Right, LEU is leucine, GLY is glycine. So what do you see? Basically, this will tell me, starting from serine, this is the entire chain ending with what? TRP. TRP is tryptophan. Okay, so this sequence, what and all are the amino acids present at what place, is what is given by your primary structures. Okay, you can understand, right? This aspargine, glycine, phenylalanine, lysine, all these things are there. The sequence, what is the sequence of amino acid present, is what is given by your primary structures. Okay, right, cool, very easy, right? So all you have to understand is primary structure gives you the sequence of amino acids. Anybody has any questions? Okay, fine. So primary structures basically gives you the sequence of amino acids. What do you mean by a secondary structure now? Right, now please understand, people, let me show you the same diagram. Right, now for all of these, right, for all of these, hydrogen bonding becomes very important. Okay, for all of these, hydrogen bonding becomes very important. Primary structures, hydrogen bonding is not important, only the covalent bonds. Right, for secondary, tertiary, and quaternary structures, you have to know hydrogen bonding. Right, because these are formed from the hydrogen bonding only. Okay, now let me write it like this. Okay, or fine, secondary structures can be of two types. Okay, secondary structures can be of two types. Now first of all, you have to understand what is a secondary structure. Okay, now please understand, people, if the moment you see that there are so many amino acids in the primary structure, glycine, proline, all these things are present. Right, now these cannot be in straight line. Now in the diagram also, if I showed you, you see, they are not in straight lines, right? They are not in straight line, right? Why are they not in straight line? Is because they need stability. Okay, they need stability. Each and every individual amino acid will interact with each other, not only through a peptide bond. Again, I'm repeating. No, amino acids will come together, make a covalent bond through a peptide linkage. Okay, and normal, you can see the peptide linkages here. C double bond O, all these things are present. CONH, all these things are present. Okay, but other than that, between two amino acids, I can have some interaction also, other than the normal covalent bond, right? And that you see is represented by the dotted line. You see a dotted line everywhere, right? This is a dotted line. This is a dotted line. So many dotted lines you are seeing, right? All these dotted lines are nothing but hydrogen bonds. Okay, all these dotted lines are there. Normal solid lines are covalent bonds, that we know already, right? But this dotted lines, wherever you see a dotted line, all these are basically going to be your hydrogen bonds. Okay, so a straight chain molecule I have like this. If I draw a diagram, then you'll easily understand, right? So say I start with an amino acid. Okay, some amino acid sequence I have. Now see that here I had say COOH, assuming, right, it is having a COOH, and say this is having OH. This is my serine, for example. Like one part of this is serine, and another part say is what is that? Any acid, any glutamic acid. Okay, one end of the chain I have glutamic acid. The other end of the chain I have your serine. This is your primary sequence. So many amino acids are there here. Okay, now what will you understand is they will not be able to stay in a straight line like this. They need stability. So what will they do is between this hydrogen and this oxygen, I can have a hydrogen bond. So what will happen now? It will shift, it will bend, all these things will happen. And say, if they bend, here maybe I had an NH2, and say here I had a OH, for example, right? Again, I can have a hydrogen bond between this hydrogen and this oxygen. Again, it is going to make it more complex, right? So many hydrogen bonds between the side chains are possible. So the ammonia, the proteins' primary sequence or primary structure bends and folds, and all these things will happen, right? Now that will end up in a secondary structure. Okay, that will end up in a secondary structure. I would say folding of primary sequence due to hydrogen bonding. Okay, due to hydrogen bonding gives you the primary structure. Folding of primary sequence, primary structure with the help of hydrogen bonding results in a secondary structure. Okay, right. Now see, for the same compound, okay, if I write the secondary structure, right? So is this the correct one? No. Okay, not an issue, even if I'm not able to show it, right? All you have to understand is they're going to fold and they're going to give this kind of structures. Let me show you how, anyways. Right. Now, I told you secondary structures can be of two types. Right. Now, they can be something called as beta pleated sheets. Right. They can be something called as beta pleated sheets. And what is the other one?
Alpha helical.
Alpha helix. Okay, alpha helix. Right. Now, this beta pleated sheets again can be of two types. They can be parallel or they can be anti-parallel. Okay, so secondary structure can be of two types. It depends on how they fold and what structures they bring into. Okay, one can be an alpha helix, right? The other can be a beta pleated sheet. Okay, now I cannot show you here because it's very complex. I can show you directly in the diagrams if possible. Let me show. just this. I'll get a better idea. Right, I'll take this one. I will. Okay. Oops. Okay, so now if you look at this, right, now you see that there is some kind of this thing you can see here, right? Or you will be able to see it in so many places, right? Are you able to see this kind of ribbon kind of structures? Are you able to see? Here also it is there. Are you able to see that? Ribbon structures.
Yes, sir.
Right. So these ribbon structures are what are called as alpha helices. Okay, that ribbon structures you see round, wounding, all again. Why this happens? This happens because of hydrogen bonding. Right. There will be so many amino acids here, people. There will be so many amino acids. Right. So if I draw a helix like this, okay, see, let me draw. Okay, now it would have been a straight line and this became like this. Why? Because you will see that, for example, in this particular point and in this particular point, I could have had a hydrogen bond. Or say in this point, this point, this point, this point, between these 2 points, I can have hydrogen bond. That is why they are mixing like this. That is why they are folding like this. Are you able to understand what I'm trying to say? Right? Why they are wounding themselves is because in suitable positions, you have hydrogen bonding possible. And that's why they shift like this. Okay. These are what are called as alpha helices. Okay. Then what I told you is about beta pleated sheets. Okay. Now, wherever you see an arrow mark, here you're able to see an arrow mark, arrow mark, arrow mark, arrow mark. Okay. All these are going to be beta pleated sheets. Okay, now if you see that there'll be 2 arrows, and if these 2 arrows are going to be pointing in the same direction, now you see that this particular arrow and this particular arrow is, sorry, I would say, right now this particular arrow and this particular arrow which you're seeing here is the same direction. So this will become a parallel beta pleated sheet. Beta sheet, I'll call it. It will be beta pleated sheet. Right. But if I consider this one, which is here, and this one, which is here, they become anti-parallel beta pleated sheets. Okay. Again, why? They are going to be formed because of hydrogen bonding. How the hydrogen bonding is formed, not important to you. Really not important. Okay.
I have
right.
a doubt, sir.
Yeah, go on.
So this is formed between 2 polypeptides, right, sir?
Yes, correct. No, need not be. Need not be. It can be in the same polypeptide that can fold and then they can have a hydrogen bond.
Within the same polypeptide also, sir?
Within the same polypeptide is also possible. Between 2 different polypeptides are also possible.
Okay, sir.
Right. I have 2 big polypeptides, 2 different proteins itself. Again, these 2 proteins can come together because of hydrogen bonding. But again, at that time, I will not call it as a helix or parallel or anti-parallel beta sheets. That there, the naming will be a little different.
Okay, sir. Understood.
Okay, right. And other than that, you see some kind of twisting and turning, some random structures are there, right? Again, those are coming from the hydrogen bonding. Now these are the primary sequences. You see a line here, right? But that is not a straight line. It is somehow folded. All these are amino acids. They are folding. This thing is happening. That thing is happening because of hydrogen bonding. Right? Not only hydrogen bonding, I can say any kind of secondary interaction. And the main important of the secondary interaction, and the main secondary interaction is hydrogen bonding. Other than hydrogen bonding, I can have a dipole-dipole interaction, dipole-ion interaction, right? All these things are possible. But the most important one of them all is definitely going to be hydrogen bonding. Okay, right? So I hope everybody understood what do you mean by an alpha helix and what do you mean by your beta sheet, beta pleated sheet, parallel, anti-parallel. Everybody understood. Okay, other than that, I have this twists and turns. You have something called as a twist. You have something called as a turn. All these things are there in the secondary structure. Okay, see, this second alpha helix is a secondary structure. Beta pleated sheet is a secondary structure. I would say this one thing I have here itself is a secondary structure. Right? I cannot call this entire one a secondary structure. Hope you're able to understand. We are going level by level. We were first so much inside that we could see what are all the sequences of amino acid present. Okay, now we come a little level higher up and we are able to see how, say, if I have one chain, another chain, how they're able to interact. That comes under your secondary structures. Okay, now what about tertiary structures then? Tertiary structures are formed by combination of secondary structures. Now you see, this I can call a secondary structure. This entire thing I have, I can call this a secondary structure. Sorry, tertiary structure. Why? Because you have so many alpha helixes, so many beta pleated sheets coming together to make a bigger structure, and that is what is called as your tertiary structures. Okay, you see, individual, this beta pleated sheet, this particular ribbon, or this can be a secondary structure. This can be a separate secondary structure. This can be a separate secondary structure, right? But so many secondary structures will come together to make a big structure, and that is what is called as your tertiary structure. Okay, I can call this a tertiary structure, but actual name is quaternary. Between quaternary and tertiary, there's only subtle differences. Okay, fine. So primary sequence means, primary structure means the sequence of amino acids. And when I have sequence of amino acids, they will bend and twist and turn to form secondary structures. Again, I know what are these, right? And so many structures, secondary structures from then and there, all these things will be put together to form a tertiary structure, right? And in simple words, I can tell that our quaternary structure is a combination of so many secondary structures. For example, you see, I'll show you. If I show you hemoglobin, right, you will understand. We know hemoglobin, right? All of you know hemoglobin. Where is hemoglobin present?
The pigment in the blood.
Red blood cells.
What is the spelling of hemoglobin? Okay, HAE. Okay, hemoglobin structure. You see, okay, this is a better one. Okay, now you see that, okay, let me actually put it in the OneNote. Oops. Okay. Okay, this is fine. Right. Now, if you look at this, right, now see, there is a alpha helix. These are alpha helixes. And maybe you're not able to see the beta pleated sheets, but you can see an arrow mark somehow, right? This is an arrow mark actually. You can also see somehow there'll be arrow marks. I'm not able to show, but there are arrow marks. There is beta pleated sheets and your alpha helixes are there. Now, this particular green one is a tertiary structure. Again, this particular blue one is a tertiary structure, right? And this particular yellow one or an orange one is again a tertiary structure. This again is a tertiary structure. Okay. Now, see, these four tertiary structures will come together to entirely mark this entire thing as a quaternary structure. Okay, this so many tertiary structures. Basically, what you understand is hemoglobin is made up of 4 proteins. Okay, it is not one single protein. It is, I would not say 4 protein, I will call 4 polypeptides. Okay, I will call them as polypeptide. There is a subtle difference between polypeptide and protein. We'll address this very soon. But for now, you understand that hemoglobin contains 4 polypeptides. Okay, 4 polypeptides will have 4 different tertiary structures, right? 4 different tertiary structures, and these tertiary structures will come together to make the entire hemoglobin molecule. And the structure of hemoglobin, I will call it as a quaternary structure of hemoglobin. That is what I call it. Right? Hope you all understand what do you mean by primary. Just the sequence, little one layer up. You see the hydrogen bonds. You see that obviously what is happening? They are twisting and turning to make secondary. Again, so many secondary structures, so many alpha helixes, so many beta pleated sheets will come together to make a tertiary structure. Again, how they come together? Through hydrogen bonds. Don't worry about it. Right? And what is a quaternary structure now? Some proteins are made up of more than one unit, more than one polypeptide chain, more than one tertiary structure. And these tertiary structures will come together to make a quaternary structure. Okay, anybody has any doubts, any clarification required anywhere? Okay, I hope everybody is following, right? So yeah, that is about proteins. That's it. This is all you have to know about protein. And yeah, what is the difference between a polypeptide and a protein? Right? Say a protein is or I would say, I'll do like this. A polypeptide becomes a protein, right, when its molecular mass is greater than 10,000. That is the difference. Okay, so basically not all polypeptides are proteins. Okay, some of them are proteins, some of them are not. Right, now how do you decide whether a polypeptide is a protein or a polypeptide? When the molecular mass of the polypeptide is greater than 10,000, then it becomes a protein. That's the only criteria you have to understand. Okay, right? When a polypeptide molecular mass is greater than 10,000, then we call it as a protein. If it is lesser than 10,000, then we call it as polypeptide only. Okay, right? Fine. So that is about the proteins done. The last part is nucleic acids. Okay, nucleic acids, right? So just because you see there is an acid doesn't mean they're acidic in nature. Okay, these are just some terms we use in biological chemistry. What is the time? What is the time? Okay, we have time, right? So now nucleic acids, we have actually 2 nucleic acids, RNA and DNA, right? Now I know the full form. It is ribonucleic acid. This is my deoxyribonucleic acid. Right, now please understand, people, between RNA and DNA, right, there is only very small difference. Only one difference is there. We'll see that later. What is the difference? But both these are nucleic acids. 90% of things are common between RNA and DNA. Not even 99% of things are common. Yeah, it's common between your RNA and DNA. Right, 2 differences, 2 major differences will be there, but most of the things which will be common for RNA and DNA. Okay, now obviously we know that these are the ones which are going to be responsible for transferring the hereditary information. Right, hereditary information. Right, now you know we can check the DNA. I will take the DNA sample of you, and I'll take a DNA sample of your father and mother. You will see that there is a match. Right, now fine. Okay, now how do we start this is by understanding what are all the components present in a nucleic acid. Okay, please understand that this is also a polymer, right? But last time in case of proteins, you saw that they were also polymers, but all of them are coming from amino acids, right? Here, what you have to understand is this polymer contains so many things, not one structure. They're going to have so many different molecules, not so many different, but say 2 to 3 things are going to be there in this kind of polymers. Okay, first one which you have to know is a base, right? Any nucleic acid, right, will contain bases. I will call them as nitrogenous bases. Okay, first thing they contain is a base, very important. Okay, next thing they will contain is a is a phosphate. Right? Phosphate is basically P double bond O. Okay, basically O minus, O minus. Phosphate will be there. Okay, PO4 3 minus is what does make phosphate. Right? So phosphate will be there. And then third thing they will have is some sugars, or I would say carbo. Okay, let me call it as sugars. Okay, so you will have a base, you will have phosphate, and you will have some sugars. Okay, now sugars, we'll come to that. There are only two sugars possible. One is my ribose, the other one is my deoxyribose. I think from the name itself you understand which sugar is going to be there for which nucleic acid, right? Ribose will be there for RNA, right? And this deoxyribose will be there for DNA. Okay, ribose will be there for RNA, deoxyribose will be there for DNA, right? And phosphate is common for both RNA and DNA. And if you look at the bases, right, for RNA, or say first for DNA, there are these 4 bases possible. There's something called as adenine, guanine, cytosine, and thymine. Right? So even give names to this. We call it as A, G, C, and T. Okay, this is for DNA. And for RNA, right, we have adenine. Everything is same. Adenine, guanine, cytosine. But instead of thymine, I have another one that is what is called as my uracil. I'll represent by U, capital U. Okay, so DNA has A, G, C, D, and RNA has A, G, C, U. Right. So you see, between RNA and DNA, there is 2 differences. One is in the sugar, right? RNA uses ribose and DNA uses deoxyribose. First difference. Second difference is in RNA, you have AGCU, that is adenine, guanine, cytosine. Uracil is there in RNA, but in DNA, you see that adenine, guanine, cytosine, and thymine is there in DNA. These 2 are the only differences between RNA and DNA. Rest all are same. Okay. Now you will see that between this ribose and deoxyribose, there is no much difference. If I show you that, let me show you. Okay. Let me show you the structure of ribose and deoxyribose. Right. Now you see what is the difference here. Like maybe if I can copy this image, or I can show you here itself. Right. Now you see this is a 5-membered ring. Ribose is a 5-membered ring. This is also a 5-membered ring. Right. Now you see at the 2' position, like you have a 2' here, 2' here. Okay. Now you see the 2' carbon. Ribose is having a OH. Ribose is having a OH. But here, in case of this 2', in case of deoxyribose, you have a H. This is the only difference, people. You see, very subtle differences. One OH you put here and you don't put the OH, the entire structure changes. Right? The entire structure changes. Right? I think you need to know what is the structure of deoxyribose and ribose. I think it will be given in your textbook also. So please know the structures of ribose and deoxyribose. It might be asked in the exam, but 90% they will not ask. But if they ask, you must be able to know. And now it is going to be only MCQs. They'll give you the structure and ask you whether it's ribose or a deoxyribose, something like that. So just know it. Just know. At least know the difference. In ribose, I have a OH, and in deoxyribose, I do not have a OH. I have a H in the 2' position. Very important. In the 2' position. Okay. Right. Now, I will show you the structures of adenine, guanine, cytosine, and thymine, and uracil. Adenine, guanine, cytosine, thymine, and uracil. Great. Google is so good. Right. Directly I have. Now look at this. Right. Now, these are the structures, and please, you do not have to remember anything. Right? I told you they contain nitrogen. They are nitrogenous bases. Right? Say all of them are going to contain nitrogen in them. That is why they are called nitrogenous bases. So you can see adenine, how it looks, how guanine looks, how uracil, thymine, cytosine looks. And we all know which 4 are present in which nucleic acid, right? In DNA, I know adenine, guanine, cytosine, and thymine are there, right? And in uracil, you will see that, in RNA, you will see that instead of thymine, you have uracil. That is the basic difference. Okay, right? Fine. Now, you have to understand how are they coming together. Okay, now I understood that there is a phosphate. There are these nitrogenous bases, and I have the sugar. Correct? Right? Now we have to understand what kind of chemical bonds, how is it that a covalent bond is present between all these 3? How are they attached? Okay, guys, just give me a moment. Give me 1 minute. I'll be back. Okay, just give me 1 moment. Okay. So now we understood what are all the different things which are present, and now we have to understand how are they attached with each other. Correct? Okay. Now let me show you. For that, I have to search it as okay. Now look at this, right? There is something called as a nucleoside, and there is something called as a nucleotide. Okay, right. Now, when you have the sugar, right, when you have the base, all the bases are 6-membered ring, people. Please understand, right? All the bases you see are 6-membered rings with nitrogen. You would have seen adenine, guanine, how they look, right? All of these are 6-membered rings. Now, when you have a bond between the 6-membered ring base and the 5-membered ring sugar, that linkage, you see a linkage here, right? Now, this entire structure is what is called as a nucleoside. Okay, now you see here also there's a nucleoside. There is this base and there is this sugar forming a bond, right? Now, if you attach a phosphate, that is PO, we know it is PO4 3-, right? If you attach a phosphate to this, then it becomes a nucleotide. Okay, base plus sugar is nucleoside. Base plus sugar plus phosphate becomes a nucleotide, right? Now, this is one thing. So obviously, nucleic acid is a polymer of this nucleotide. Again, I repeat, nucleic acid is a polymer of nucleotide. There will be so many nucleotides like this, which will be coming together to make the entire nucleic acid. Okay, now if I show you another diagram, that will make you a little more sense. I need to show, yeah, this is a better structure. Now you see, this is a nucleotide, right? Now this is your, okay, looking at this structure, it is a ribose or a deoxyribose, guys? This one, is it a ribose or deoxyribose? This particular 5-membered ring, the sugar I have, is it a ribose or a deoxyribose? Are you guys there?
Yes, sir.
So what is this? Is this a ribose or a deoxyribose?
Deoxyribose, sir.
Now see, here I have a OH in 2' position. I have a OH. If I have a OH, what is it called as?
It's ribose.
Ribose, right? If in this position I had a H, then it becomes DNA, right? So this particular molecule, what we are having here is for RNA, one particular nucleotide. Yeah, one particular nucleotide. You see there is a phosphate, right? Phosphate attached to the sugar, and sugar attached to the base. Very simple. Right? Sugar attached to the base. This is the functional unit of any nucleic acid. Right? And this is adenine. Okay. This is the structure. If you're wondering what is this is adenine. Okay. So this is how the backbone of nucleic acid will be there. Right? And moving on. So now we understood what are all the components present, right? And how can there be any kind of nucleoside or nucleotide, right? Okay. And I want to concentrate on this particular one. PO something is there, right? Okay. This phosphorus attached with this sugar, right? Now this linkage, see, for a polysaccharide, we said that the linkage was glycosidic linkage. For a polypeptide, we told that the linkage is a peptide linkage. Same way for a nucleic acid. And that too in a nucleotide. Nucleotide means basically the phosphorus plus the sugar, right? Plus the base, right? I am talking about this particular bond. Right. Now, this particular bond would be a phosphodiester linkage. Right here, between the phosphorus and sugar, right, what kind of linkage you have is what is called as phosphodiester linkage. I think I can show you better. I can show you a better diagram in net for this phosphodiester linkage. I need to see how to amino wait. Oh. Now you see, right? Do not forget about, I mean, look at the end diagram only, right? Now you see this particular, let me actually copy this image. Right? Oh. Okay, so you really don't have to worry about this part, forget about it. Now look at this part alone, right? Now you see there is a sugar. No, this is a base, right? And this is my sugar. Now look at the sugar and tell me whether it's ribose or deoxyribose.
Deoxyribose.
This is deoxyribose. You see that there is a hydrogen, not a OH, right? This is a deoxyribose, so this is for a DNA. Okay, now you see that there is a base. Forget about what is the base. And you see that there is a sugar, and you see, I would say what you have to understand is a nucleoside and another nucleoside, right, will be linked through phosphorus. Okay, this is one nucleoside, people. Base plus sugar, is it not? This is base, this is sugar. This is one nucleoside, right? And you see that there is another nucleoside, right? Now these two nucleosides are together bonded with the help of this one, which is my phosphodiester linkage. Okay, and this entire thing along with phosphorus, if you take, that becomes a nucleotide, right? Again, this one along with the phosphorus, if you take, this becomes one nucleotide. Right? Now, I have two nucleosides. Two nucleoside means what? Nucleoside means the sugar plus the base. Sugar plus the base. Two nucleosides are attached together with the help of this phosphorus. And the linkage you see is what is called as your phosphodiester linkage. Okay, I don't think you need to remember the structure. No structure you need to remember. I'm just showing you how it actually forms. Okay.
I have a doubt, sir.
Yeah, go on.
This only occurs for a nucleotide, right, sir?
Yeah, no. So basically, this is one nucleoside. One base, one sugar is nucleoside. And again, one base, one sugar, nucleoside. Right. So one nucleoside, another nucleoside is combined with the help of this phosphodiester bond. Right. Now, along with the phosphorus, if you're going to consider that is the base plus sugar plus phosphorus. Oh God, 1 second. Right. So if you have base plus sugar plus phosphorus, this entire unit, if you take that, is going to become a nucleotide. Okay. So the question, how should I properly put it is two nucleosides, base sugar, base sugar. Between these two, I will have a phosphodiester linkage. That's it. What do you mean by phosphodiester linkage? I'm going to have phosphorus, one oxygen, another oxygen, combining these two. That's it. That's exactly what you're seeing here. If I can just rub everything and show you.
Sir, I have a doubt, sir.
Yeah, go on.
Sir, but it's not like you're introducing phosphorus, right, sir? But it's already present in the compound.
Yeah, it's 100% in the body. Yes, absolutely. Yeah.
So how can two nucleosides form a bond? So it'll always be either between nucleotide and nucleoside or between two nucleotides.
Correct.
Right?
Two nucleosides cannot form a bond. Nucleoside, what do you have? In nucleoside, you just have the sugar and the base. Right. Again, this is a nucleoside. This particular structure is a nucleoside. Right. Now I'm telling you, two nucleosides can be attached, can come together with the help of phosphorus. And that is nothing but your phosphodiester bond. Are you able to get the point?
Yes, I understood now.
Right? Okay. Anybody has any other questions? Okay. And none of these you have to remember. You just have to remember nucleoside means what? Nucleotide means what? What are the bases present? The names of the bases and this linkage name, phosphodiester linkage or phosphodiester bond. Only the names you have to remember, people. Nothing else. No structures. For your level, no structures. Okay, and one last thing I will tell and I will conclude. Right now, please understand that DNA is a double helix. Right, DNA is a double helix, but RNA is a single helix. Okay, DNA is a double helix, but RNA is a single helix. Okay, what do you mean by double helix? Now say RNA single helix means it'll be like this. Helix, normal helix. Okay, something like this. It will be a two line. It will be a two parallel lines going like this. Okay, please bear with my diagram. Okay, this is going to be something like this. Now, if you look at DNA, it is going to be a double helix. Double helix. Please bear with my diagram, people. This is one helix, right? And there is going to be another helix. Again, that is going to be something like this. Okay, so one helix, two helix. See, basically these are the chains. Sugar, base, and phosphate. Sugar, base, and phosphate. Sugar, base, and phosphate. This will be in one helix. Again, another helix will have the same thing, right? But you will understand that between these two helixes, there is going to be hydrogen bonding, right? Between the two helixes, right? Between the two helix, you are going to see hydrogen bonding, and that is why they are able to be together. Okay, now please understand, for hydrogen bonding happening in DNA, right? All you have to understand is A binds with C, right? Am I right? Yeah, A with C and G will bind with T. Okay, AC will have hydrogen bond, right? And G and T, guanine and thymine, or U, basically either G, T, or U will have a hydrogen bond. Okay, now if I can show you that, Right? I hope you all understood, right? DNA is a double helix. Double helix is found because two individual helix are bound together with the help of hydrogen bonding. Okay, but in RNA, generally, it can be a double helix sometimes, but most of the time you will see that RNA is going to be a single helix. Okay, right. Now let me show you how exactly the hydrogen bonding is formed. Again, this is not important, people, just for you to understand. Okay, so this is what is called as Watson-Crick base pairing. But again, you do not know this Watson-Crick base pairing and all. Okay. Oh, I'm sorry. It is A binds with T. I'm so sorry. Let me have to change this. Right. I'm sorry. This is entirely wrong. A binds with T or U. Right. And A with T, hydrogen bond, and G with C, hydrogen bond. Okay, fine. Let me show you how this is formed. Right now, do you see? Right now, this is my adenine. This is my thymine. You see that there's a dotted line. Right? I have a hydrogen bond. Same way between guanine and cytosine, you see that there are 3 hydrogen bonds. This is very important. Okay, between guanine and cytosine, there are 3 hydrogen bonds, and between adenine and thymine, I have only 2 hydrogen bonds. Okay, adenine, thymine, 2 hydrogen bonds. Guanine, C and G will have 3 hydrogen bonds. Okay, I don't think that is important for you, but yeah, you can understand that because of this hydrogen bonding, say what you have to understand is this side will be one helix. This side will be another helix. Okay, right? So obviously they will interact. The 2 helix will interact with the help of hydrogen bonding. Right? If I can show a better picture with the double helix kind of formation, then it will be very good. See. I have to make this big. Right? Now look at this. I have, this is one helix. You can see the sugar, phosphate, and the base. Everything is there. The bases are here. C, G, A, T, all these things are here. This is one helix. There is another helix, right? Now these 2 helixes are interacting because of hydrogen bonding. This is one separate ring, right? This is one separate helix. This is another separate helix. Now you see C and G has 3 hydrogen bonds, A and T has 2 hydrogen bonds, 3 hydrogen bonds. Like this, you can easily have the 2 helixes interacting with each other, making it a double helix. Okay, now these kind of things might be possible in RNA also, but there it is not that prominent. But in DNA, it is very prominent, so that it is going to form a double helix and not a single helix. Okay, and we are done with this chapter. Okay. That is about DNA, RNA. Right now, any questions anybody has? Very interesting, right? See, simple chemical organic compounds. You are seeing how they are present in our body and what kind of roles they are playing. It is very fascinating. Okay, it is very, very fascinating. Right? If you like biology and chemistry, best combination you can have. I hated biology at a point of time, but when I started studying about these stuff, I looked at biology from a chemist point of view. Everything is basically a chemical, everything is an atom, everything is a molecule, and everything is a reaction. Why will I not look into that picture? Why will I not look into that viewpoint? Is what made it very interesting for me. Okay. Anyways, the last part of the chapter is going to talk about vitamins. I will just take 2 minutes, people. Vitamins, we know that different deficiency of the vitamins will have different things. Vitamins can be 2 types. All of them, we know that it is going to be fat soluble vitamins or water soluble vitamins. Right? We know vitamin A, vitamin B, vitamin C, some vitamin, whatever vitamins you have. Right? Now, what are all the fat soluble vitamins? There are only, I think, 4 of them is going to be some water soluble, or I think it's vitamin A, D, E, and K. All these are water soluble or fat soluble. I forgot. Anybody remembers?
I think vitamins is deleted, sir, from our syllabus.
Yes, vitamins is deleted from our syllabus.
Oh, then great. Wonderful. Then we don't have to study it. Okay, done. So anyways, you can know it can be either fat soluble vitamins or water soluble vitamins. Right, that's it. The chapter is done. Okay, So for new people, I think this is the first class you might have attended for me, right? I hope you all liked it. I hope you all understood. I mean, liking is secondary. Whether you understood, whether you got the science behind this is the most important thing. Okay. Any particular question anybody has? Okay. So if that's the case, yeah, we'll stop right here. Okay. And we'll meet in another weekday. We will discuss your question paper. Like that's very important, right? And we have to do the previous paper also. Right. So in the weekday, I'll speak with Raghav and I'll let you know. They will let you know when is the time, when is the day. We'll have a paper discussion. Okay. So, but anyways, from next week, we will have only paper discussion till your class because now we have completed the chapter. Okay. So next week, Sunday and Saturday, Friday, we have two more days, right? Friday and Sunday, because Friday classes also I'll take now. So we have 2 more days. Other than I just need 1 more day. Only on this week, so that we'll be able to do the paper discussion. Okay, we'll anyway speak with Raghav and let you know whatever it is. Right. Thank you so much, people. We'll meet again.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, everybody. Thank you.